So I guess we can begin. So it's a great pleasure to introduce our next speaker for the APGC Seminar series, who's Darren Wells, who's based at the University of Nottingham, where he's a Principal Research Fellow at Sutton Bonington campus, where he's and also co-director of the Hounslow facility, which many people know is a world-famous below-ground phenotyping facility. Based in Nottingham. I also know Darren through his work as a leading light of Phenome UK, which is a BBSRC project which aims to sort of scoping exercise that aims to sort of bring together different players in UK phenotyping to allow access and essentially to improve processes between us. That's a fairly poor summary. I'm sure you'll give something in your presentation, Darren.
Oh, but I've kept this Phenome UK free. This is
have you?
Going to be research. Yeah, exactly.
So if anyone wishes to look at Phenome UK, feel free to.
Actually, I've got one slide right at the end about the conference, so
okay.
people can link out to that if they want to check. But I'm happy to answer any questions about it as well at the end.
Right. So I'll be quiet now, Darren, and let you get going.
Right. Let's share the screen. I'm hoping everyone can see that.
Yeah, we've got it, Darren. Yeah.
Great. I'll move you over to the site. Right. Thanks very much, Rob. And as Rob said in the introduction, this is all going to be about what lies beneath. I'm going to talk about plant roots, why they're important, why and how we should study them, and then give an example of how we've used some of the techniques I'm going to introduce to look at the plant roots of an important crop species in Africa, pearl millet. So, tiny bit of background. As Rob said, I'm based at Sutton Bonington. We're about 8 miles out of Nottingham itself and the main campus. This is us from the air. All the work I'm presenting today will have been done in plant and crop sciences, these two buildings that you can see the little red circles around, and the Hounslow facility is the one on the right. I'm currently sitting on the one on the left, plant and crop sciences. And as will become clear when I'm talking about a lot of the work we do a lot of collaboration with computer scientists to do some of the analysis side of our phenotyping. And all that happens on our other campus, it's called Jubilee Campus, and again, 8 miles away in the centre of Nottingham. And this is us, we're one of the last few, like you, we have our own research fields and research farm. Unfortunately, we're also right next to East Midlands Airport, so we don't get to fly any drones over them, but we do a lot of our phenotyping out in our own fields as well. But overview of today's talk, I'm going to talk about why I think we should study roots and study roots more. I'm going to go through the various techniques that have been basically developed over the last 10, 15 years. There's been a big increase in the techniques designed to study roots, particularly roots in situ. And there's a lot of new technologies I'll talk about. And then I'll show how those are used in one particular case study, which is this work on pearl millet. So why study roots? As we teach all our students, that there are 4 things that we drum into them that roots are for. They're important for water uptake, nutrient uptake, for anchorage and storage of water and carbohydrates. Of course, there's also a lot of specialized functions on top of that. Anything exposed to evolution will evolve to suit different tasks. And the mistletoe showing there a bit of parasitism. Roots do lots of other things other than those top 4, but those top 4 are the things that are mainly important to plants. And there's a place for roots and root research in addressing one of the big challenges of our age, which is global food security. And the FAO's definition is the one I tend to use, is this one, which is food security is all people have physical and economic access to sufficient, safe and nutritious food to meet their dietary needs. That's difficult anyway, but it's harder in our particular next 10, 15 years, is because there are a lot of threats to global food security. And if we look back at where we are now, the ability to feed the population we have came from the Green Revolution. Late 50s, early 60s, we managed to triple food production in that period up to the 1990s, based on 4 main core technologies: an increase in mechanization, the use of widespread use of chemical fertilizers, new classes of crop protection chemicals, and from a plant biologist point of view, improved crop varieties. And Norman Borlaug, of course, was the father of those new crop varieties, particularly dwarfing. He got the Nobel Peace Prize, and quite rightly so, in 1970 for the work he did introducing dwarf wheats. And dwarfism gives you lots of advantages. The stems are shorter, which means more of your carbon goes to the grain, so you end up with a higher yield. The fact that the stems are shorter means that it can survive one of the effects of chemical fertilization, which is bolting. And so it prevents lodging due to bolting, which means it underpins one of those other pillars of the Green Revolution, the chemical application of fertilizers. Also, shorter stems are easy to harvest, which means it met that other pillar of the global root of the revolution, the idea that you can increase mechanization. Put all those together, and as that diagram showed, we managed to triple yield over the period we were talking about. And we now know the genetic basis of a lot of that dwarfism. It's the RHT genes, and this has been used in many other species. The problem and the thing facing global food security is at the peak of the Green Revolution, we had about 3 billion people on the planet. Today, we're looking at about 8 billion. And those 8 billion people have also been fed by that tripling in production. In the next 25 odd years, the latest estimate is we're going to go to 9.2 billion. It's dropped a little bit. My older version of this slide had 10 billion. There has been a reduction, but it's still an extra 3 billion people to feed with what we have. And there's the problem. This is the UK version of that graph I showed a couple of slides ago. Our wheat yields, we can see in the period from 1960 to about 2000, we've got that great increase. We went from 2 tons a hectare to about 8. But it's flatlined since. And it's not only flatlined, but it's a bit variable year and year. And that's possibly an effect of climate change and the fact that we're seeing in yields wildly shift from year to year. So we're not increasing our yields, and yet we've got to feed another 2, possibly a bit more billion people. This led to the concept that because the Green Revolution mainly concentrated on what people could measure, which are above ground traits, is everything going underground an unexploited resource? And there's a little bit of evidence for that. So the Waynes and Eadie study 2007 looked at the parents of some of the Green Revolution early lines, and their root biomass was about 2/3 the size of land races. So the land races definitely had larger root systems. A slightly contrived image on the side shows graphically the difference between ancient and wild races and cultivated wheat. And so this led to the concept that root traits have been overlooked, and there's possibly something back in the wheat ancestral tree that we can breed back in, and there are root traits that we might be able to use to get that increase in production. Jonathan Lynch coined the phrase the second Green Revolution. He concentrated very much on root architecture, but there are other things in root function that this second Green Revolution could deal with. And he posited the idea of there's a bunch of optimized architectures that will help drive an increase in yield. You could imagine things like more roots, longer root hairs. A deeper root system would have more advantage in getting things like water and nitrate that tend to be deeper in the soil profile. A shallower root system would work if you were looking at getting things like phosphate and micronutrients that tend to live in the topsoil. There are anatomical and physiological traits for the functioning of the root system that could be improved. And the idea that what we really need are lines that are resilient, in fact, to this changing climate, and the fact that we're not seeing the same climatic or even soil conditions from one year to the next. And can the root system help even out yield over multiple years? And the reason there wasn't a lot of studying on the root system back in the 50s and 60s is because it's difficult to study. Soil is opaque, we can't see what's going on. And that leads the techniques to be divided into 2 very clear classes, those that are invasive and destructive of the root system. You see a picture of an example here. This is a root system being yoinked out of the field. That's obviously never going back in again. That's a destructive technique. And on the right-hand panel here, you can see an MRI scan of a root showing scans done in situ. So a very broad classification of how you can do this kind of thing is to be destructive or to be non-destructive. Irrespective of the nature of the technique you're going to use, that there is a need to increase throughput. A lot of these techniques are inherently low throughput, and you can do that either by automating how you take your sample or how you image it, or you automate how you deal with the data once you've got it, how you extract the data from whatever your system is. And what I will say here, and nature in all of the techniques I'm going to talk about over the next 45 odd minutes, are that these techniques are often complementary. There's a definite balance between the number of samples you can run in a system, what developmental age it is, what conditions you're growing them under. And there are, as you'll see in the example I give at the end, there are definite benefits in using 1, 2, or 3 of these systems at a time and exploiting their complementarity. So the destructive techniques, the simplest thing is to simply dig up a root system. This had the advantage you can do it in the field. You can just walk out to your field at a certain point and either dig a trench or dig out the individual root system. You can do a similar thing if you're growing in controlled environments in pots. You can grow your plants in a pot and wash off the soil. And there are systems that have been in place for quite a long time, programs like WinRhizo using flatbed scanners to extract data from washed root systems. The problem with these systems are that you need, in the washing from soil for small plants, you lose the structure. You lose a lot of the architectural traits that might be useful. A way of slightly less damage to your field than digging up all your root systems is to use soil cores. And you can use soil cores a bit like the washing system, where you wash the entire core and extract the roots at each profile, or you can speed it up by doing core breaking, which is what this little image shows. And this was a system developed by Michelle Watt's group that uses core breaks. So you break the core at certain places and then simply quantify the number of roots visible at that break. And they had a nice system with a UV camera where you could image quite a lot of cores at a time. So increased throughput by adding a sort of semi-automated step. But the problem with this is you are only extrapolating from that core to what's happening to the root system for each individual plant or group of plants. If you want to look at the internal structure of a plant, the anatomy, the arrangement of tissues, you need to move definitely to destructive techniques. None of the important species that we work on you can live image particularly well on things like confocal microscopes. Unfortunately, they're not like Arabidopsis. So if you're doing sectioning or laser ablation, which I'll talk about a little bit later, that is entirely a destructive technique. So it's a quick overview of shovelomics. It literally, the word describes what it does. You dig up the crown of your species of choice, wash it, and then the original system had a little chart where you laid the crown over and then you simply scored it against angles of various root systems. You can increase throughput in that kind of system by, instead of using a scorecard, take a picture and then develop some image analysis software. This is Tino Columbi's work, where he had a little bit program that helped you take pictures and then extract some root traits from those crowns immediately. There's another system, again, that came out of the Lynch group that can do something similar. If you're not going to dig up plants, the other way to do it is to use artificial media. Oh, sorry, I meant to say I should have introduced this section as the non-destructive techniques. If you want to leave your plant in situ, which gives you the advantage of the root system will maintain the shape it probably has in the soil, you can use entirely artificial systems, transparent media. For that, for a long time, agar has ruled the roost, but there are other transparent gels you can use. Hydroponic systems, Naphion acts like a kind of transparent substrate, and glass beads have been used. This gives you the kind of the structure of soil or of something soil-like, but because it's transparent, you can take pictures of it. You can use risotrons in various flavors, risotrons, risoboxes, risotubes. These are simply a soil container which has one or more transparent window, and then you image through the window. This has the advantage that you're growing your plant in soil, but you're only being able to visualize the roots that intercept the glass window. So you're looking at a subset of the root system. If you truly want to see the root entirely surrounded by soil, as it is in nature, you need to move to the various tomographies. And the 3 players in this field are X-ray computed tomography, magnetic resonant imaging, and positron emission tomography. And I'll talk about those a little bit later. And the final thing you can non-destructively do in fields is to use two techniques from geophysics, electrical resistance tomography or electromagnetic inductance. And these look at roots via proxies. They effectively can map water distribution in the soil, and then you can relate the water distribution to where the roots are. It's not as good, but it's non-destructive and works at the field level. In a similar way, you can use neutron probes to track water and then correlate your water to your root systems. So these are some of the systems we developed at Nottingham over the last 10 years or so to deal with, firstly, automating the acquisition in transparent media. So these are Arabidopsis plates being imaged by little robot systems that move up and down growth room plates. And with each generation of the robot, we turn to our computer science colleagues and ask them to help us develop software systems. So we have a software system called Root Trace, and the second version of it, Root Trace 2, that automatically deal with the images that come out of this kind of system. Because as soon as you increase the throughput of your imaging, your bottleneck becomes image analysis. The latest version of this system is much cheaper, much faster. The technology has moved with us, and we can now build this entire system for a fraction of the price of those first ones. And as you can see from this video, it's in real time. It's moving very fast. And the kind of work you can do with this kind of thing, when you can screen in these numbers, you can screen large populations. So we recently published with Jérôme de Pessemier some work on some natural accessions of Arabidopsis, looking at the effect on root system architecture of nitrate levels. And if you want to build one of your own, the Bagley paper I've referenced there has all the plans to build this system. Moving up slightly to scale, if you want to do things like wheat plants or rice, barley, it's a little bit too big. You won't get much growth out of an agar plate. So we moved to a hydroponic system. This is a hydroponic system using germination paper. So we use plant-safe germination paper from Anchor. These are racks that will hold 600 plants in a standard growth room in 6 tanks, about 100 plants a tank, which means you can do different treatments. And good for numbers, but the problem with this kind of system is it's very hard to automate. We can't use our automation route here to increase the throughput. This is very much somebody has to be there to move the plants out of the tray to the imaging station. So we concentrate on the automation of the image capture itself, and we do that with barcode readers, automatic imaging. So that speeds up that step, but that is still a semi-automated system. Similarly with the software we developed for that is called RootNav. And as you can see from the two pictures on the right-hand side, it extracts all the root data that we can think of at this time. It fits a spline to each of the root, it works out the root classes, and it works out things like angles and areas covered classes. The problem with that system is at the time when we wrote it, it was very hard to fully automate it. So it's what we call user assisted. So the user has to come in and tidy up the image a little bit to make sure it's found all the right parts. An advantage of having a manual system first, or a semi-manual system, is that you quickly develop a large data set of expert annotated images. So John Atkinson, who's the author of the paper in 2015 that used this system, had manually or semi-manually used the RootNav program for something like 6,000 or 7,000 images. So with that number of images, you can train deep learning systems to do it for you. And so we fed all of the data from John's work in 2015 into a neural net program, and we immediately got something that is 99% there for every root in about 5 seconds a root. And the power of some of these deep learned models is that you can almost reverse engineer them. So we taught it how to do wheat from all the wheat images from John's PhD. And we then went back to our Arabidopsis images and said, can it do architecture on plates of Arabidopsis? And the short answer is yes, it can. And so now we have a fully automated system that will automate both our plate images and our images from the 2D system. And this is what you can do with that kind of system, going back to that idea that there are root traits that we bred out prior to the Green Revolution. This is Glasgow, which is no longer a commercially grown wheat, but was a few years ago, and various members of the wheat family tree. And as you can see, the output of the program is showing us there are measurable differences in the root system between the ancient relatives of wheat and wheat itself and modern wheat. And so the obvious use for this kind of system is to look at mapping populations. So I've introduced John's work. John looked at the Rialto Savannah double haploid population. And because we have the full sequences of the population, because we have this phenotypic data from those 2D experiments, we could go hunting for QTLs, and we have a very strong candidate on chromosome 6D for early vigor. In these seedlings, it seems to promote larger, faster growing root systems. Later experiments using the same system by Marcus Griffiths in my group were looking at the effect of nitrate on this double haploid population. And again, phenotyping using this system, we have a bunch of candidate QTLs for N-dependent genes and N-independent genes. And interestingly, the genes that you can just see in green on the right-hand side on 6D are N-independent. So that's the same QTL that we found in the non-N treatment, which is quite an interesting validation of that. This idea of complementarity, the idea of proving something works in multiple systems, carries on here. So we took those same lines with and without this QTL on chromosome 60, put them into a rhizotron system. You get fewer sample numbers because we've only got about 30 rhizotrons in this setup, but you're seeing them in soil. The images are also harder to analyze because the soil is getting in the way of the segmentation, but you can see those lines with this QTL are growing larger root systems than those without. So that's a validation in kind of what we found in the seedling screen. While we're on the subject of rhizotrons, these come in many shapes and flavors. There are rhizotubes, which are effectively a kind of combination of that germination paper system and soil. So the soil sits inside the tube, the paper goes around the outside, and then you can put these on conveyor belts and image them in large numbers in glass houses. And field mini rhizotrons are basically perspex access tubes through which you drop either a camera or a scanner. And these have the advantage that they're you can put in multiple tubes but only have one scanner system, so you just walk it around your various tubes. The disadvantage is, again, like the soil cores, you're then extrapolating those routes that intercept your imaging tube to what's going on in the bulk soil, and it may or may not be a good system. But they are in soil and they're not disturbing that much the roots around it. So moving on to non-destructive techniques with full imaging, these are as I mentioned, the tomographies. So PET is using gamma rays and radio tracers. MRI is based on water content, it's magnetic fields, and it uses radio frequency waves. And the technique we use here is X-ray microcomputed tomography, which produces a density map based on the attenuation of X-rays through the sample. And each system has its advantages and disadvantages. MRI is particularly good because the resolution is nice, fast acquisition time. It works in very wet soils. It doesn't care how wet the soil is. The drawback of MRI is they generally tend to sieve the soils, treat the soils, and remove all the paramagnetic particles. And that could be a problem if you're interested in a particular soil with particular features, and the paramagnetic particles are the ones you're interested in. So it does require a semi-artificial soil. PET is great for active following of tracers, but its resolution is low. And so a nice usage of combining these technologies is there's often MRI machines and PET machines in the same place, particularly in hospitals. And if you can borrow time on them, you can do MRI and PET almost simultaneously. So you do an MRI scan, then you take the same pot and you feed it something like 11C labeled carbon dioxide, and then you follow where the carbon goes by looking at the tracer signal. You overlay the two, you see where the tracer is in the root system. That's a very powerful set of techniques, but again, not particularly amenable to high throughput. The technique we use at Nottingham is X-ray CT, which works very well in soil. It's again relatively low throughput, but if you are going to move it to a high throughput system, you have to purpose build your technologies together. So this is our purpose-built centre, the Hounsfield Centre, named after Sir Godfrey Hounsfield, the guy that got the Nobel Prize for CT machines. And as you can see from this, it's a big shed. That's where our CT machines live, and a glass house attached to it is where we grow our plants. We have 4 CT scanners inside the Hounsfield. This is the one that does the work attached to the glass house. As you can see, we've automated moving of mesocosms containing our plants into the machine. We do this because A, they're very heavy. They're about up to 70 kilos each column. And B, the cost of this machine, to increase the throughput, we want to run it 24/7, 7 days a week. And we can do that using this kind of industrial level of automation. Throughput is still relatively low because we can only fit, with that automation system, 140 plants in the glass house at any one time. So compared to a big field trial, it's still a relatively small number of samples. But we get really useful information out of it. So these are, again, the ancient relatives of wheat. And we can see in soil, in meter-long columns, real topsoil, we're still seeing that difference in root systems architecture that we saw before on the seedling screens. So we can start to do some really interesting things now with roots in soil using CT. And some of these mesocosms, become very interesting experimental chambers. So we can look at root-root interactions. And again, this relies heavily on software. Stefan Meyerhofer from the computer vision group at Nottingham developed tracking software that lets you disentangle 2 different root systems together when they're in the same pot and they've been scanned by CT. So here we look at 2 wheat plants growing together and wheat and black grass in the same pot. And this is going to be really interesting to look at inter and intra-species differentiation in root systems in soil. But possibly the most important aspect, and the thing that we know less about, are root soil interactions at this level. And we can use CT to look at the same time we're phenotyping for the plants and the root system, we're effectively phenotyping the soil. And a lot of the work in Hounsfield is about phenotyping soil. It's looking at things like the effects of compaction, tillage, practice, effect of the microbiome on soil structure, that kind of thing. And this was a nice experiment from a year or so ago that was taking field cores and scanning them and looking for where wheat roots had grown and the relationship between the pore structure of the soil and where the roots end up. And probably not surprising, at a certain size, roots will grow down biopores rather than grow through the root system itself. And that, of course, has an impact when you're inferring things like root shape and root distribution in space, because if that's dictated by the biopores, it's not finding its own angle. It's going to take the path of least resistance. So it's always important to phenotype your substrate at the same time. A system, you don't see this in the 2D systems or the artificial systems, but you will see them in the real system. And one of the things I'm interested in is moving downscale one more, is to look at the anatomy of the root system itself. And again, it came out of the very prolific Lynch lab. They coined the phrase anatomics. It's effectively phenotyping for anatomy at high throughput. Specifically, they talked about field-grown plants, but we'll do the anatomy of anything. And you can increase throughput of this by using new technologies like laser ablation tomography, which I'll talk about in a second, or by adapting existing techniques. And myself and John Atkinson a few years ago, before we could afford a laser ablation tomograph, worked out a way to increase the throughput by using confocal microscopes for fast, accurate imaging and vibratomes and embedding systems. You can do some very good work on anatomy with equipment you already have. That said, laser ablation tomography is a step change in our ability to look at plant anatomy at high throughput. Very simply, it uses ultra-short lasers to ablate material without introducing much heat damage. You then link that to an imaging system and you can take very fast 2D and 3D sections. We were very lucky to get the first tomograph outside of the US, funded by the BBSRC Alert program, and they are just about becoming commercially available now. So this is the laser ablation tomograph as it arrived. This is the commercial version that you can probably now buy, though I don't think many of these have been delivered yet. As you can see, our model, I suspect our model was a bit of a test run for the spin-out company from Penn State that sold it to us. Because of who we are and how we build our phenotyping systems, the first thing we did to the tomograph was adapt it. We changed the optics, we changed the software, we built a few safety features. And the reason we did that is because we were interested in very fine detail. So the way a laser ablation tomograph works is on the right-hand side of the image, you can just see a silver box, which is the laser. The laser comes through the galvanometer, which is the thing you can see with a lens in the top of the image. The galvo forms the laser into a sheet in front of the sample, and you simply move the sample using this blue thing with chevrons you can see on the side, which is a high precision, it's a nanometer accurate stage. You drive your sample into the laser beam, and every time it hits the laser beam, it removes that material. And because we're doing this at picosecond pulsing, it removes it with introducing very little heat. So effectively, we're just very rapidly sectioning our sample. And we swapped to microscope objectives. These are long working distance infinity corrected lenses hooked up to a machine vision camera, which means we can alter the scale at which we're doing this work. And this is shown in this image here. As with all systems, the higher your resolution, the smaller your field of view. So at our maximum resolution, which is about 0.2 of a micron per pixel, we're only seeing 0.7 by 0.4 of a mil. But as you can see, we can see some very nice structures. 10 and 5 are probably our most common magnifications that we use depending on the size of roots we're looking at, but we can go all the way up to a single times objective, which lets us do very large plants like 16 by 11 mil, which is quite large. I'll probably skip over this bit because this is a bit not less interesting, but it's interesting in terms of how the system works. You can either ablate samples fresh, or you can preserve them in ethanol, which is normally what happens where they're preserved from field sites and then sent to us. The preserving in ethanol gives us the advantage of using a technique called critical point drying. Critical point drying was designed for electron microscopy, and it exploits the fact that at the supercritical point, a liquid can go from being a fluid to a gas without going through a phase barrier. What that means is you don't get the damage associated with going through the phase barrier. You don't get bubbling, you don't get rippling effects. And because our samples are already in ethanol, ethanol is the exchange liquid for CO2 in commercial critical point dryers. So we're already in the substance that we can use to swap over to CO2 at the supercritical point and critically point dry our samples. And that you can see in this image set of images here. This is a sample that's just in 70% ethanol, and it's been put through the lat. This is one that's been dried. You lose a little bit of some of the color information on the signals, but you gain a lot of contrast, and you gain a lot of features that you couldn't measure accurately before. So we tend to routinely critical point dry, partly because our samples are preserved anyway, and partly because of this increase in resolution. So this is a lat in action. Every flash you're seeing this is a real-time image, by the way, is a fire of the laser, a taking of the image, and then the sample moves forward, in this case about 10 microns a slice. We've wrapped it all up in a fairly user-friendly interface, so with relatively a short training period, most people can use this machine. And this is what the image looks like, and the actual images themselves rather than the output of the machine. And as you can see, we've got lovely definition of the various cells. At these 10 micron steps, you can see the cell walls, the end walls are coming in and out as you dive into the sample. We saw a lateral root appear there. We're getting these root hairs appearing on the side. And this whole thing is acquired in a matter of minutes. So you can do hundreds of samples a day. So we've achieved our throughput. And the kind of traits we want to measure in this system are the things you can see. At the whole root level, we're looking at the size of the root, the relation of the steel size to the size of the entire root itself, the percentage of the root area that is cortex. And at the individual tissue level, we're identifying the individual tissues, the epidermis, the endodermis, cortex again, metaxylem, Vascular bundles, it's quite difficult to tell the difference between xylem and phloem on some of these samples, but generally it's half the number of vascular bundles for each class, so that's quite easy to do. And there are traits that appear over time, things like erenchyma that we can also trait for. So as with all our systems, knowing what we want to see, getting that data out of the images is actually quite tricky. First thing we did was use a program we developed for confocal images called CellSet, which lets us create a mask of an image and then segment out the pieces within inside that image. And then you can manually sit down and say, right, this is my epidermis, this is my endodermis, this is my cortex, mark up the various tissue layers that were on the slide before. Then you can give this to a machine learning program to try and see if you can do it automatically. And it's quite tricky. This annotation step takes quite a long time. So we have a relatively small training set, but we do have a system that works really well. So taking an old program or an older program from Penn State called RootScan, we moved that onto a machine learning version with some funding via the DARPA program in the US. And so between us, we developed a new version of RootScan, and we developed a newer version of that further, that we've moved from being something that will only run on a cluster of GPUs to something that we can run on a single computer. So we're now running this on a single computer in the Hounsfield that sits in the same room as the LAT. And this is how it works. This is a cross-section. This is Pearl Millet, and that's the RootScan output. So on a nice image, it produces pretty much exactly what we would produce. There's a tiny error. You might see a little bit of orange there outside in that cortical sclerenchyma that isn't actually cortical sclerenchyma, but we can sort that out in the measurement phase. The nice thing about deep learned models is they can surprise you. So terrible looking images like this one, it seemed to segment quite well. There's definitely errors there, but there's some recoverable data in an image that normally I might throw away. Doesn't matter if the root is broken in handling or not, the program will still happily measure it. And that brings on nicely to how we then turn that information into numbers, because that's what we want for our genetic studies further down the line. And the simple way is to count the pixels of each color, and then that gives you area and shapes for class. But you do get an error caused by what's called the point spread function. These black areas aren't really black areas. They're just saturated parts of the image. So we have a second measurement system where we use convex hulls, we set geometrical shapes around things, and then we compare the two numbers for each tissue layer, and it flags up when something's gone wrong with the system. So with that, we get quite a robust way to measure our traits of interest. So now, for the next 20 minutes or so, I'd like to talk about how we use all these techniques. And a good example project of that has been work we've done over the last few years on pearl millet. This is pearl millet growing in Senegal, Pennisetum glaucum. It's incredibly important in West Africa. In Senegal, it covers 64% of the land under cereal production. It's used for animal fodder. It's used as a human foodstuff in flour. They brew some beer from it. And it's incredibly important in the region. It was domesticated about 4,500 years ago. This is a map of the domestication of pearl millet. Senegal, you can see, is right on the coast there, sitting out. It's a little thing that looks like a face. And that whole area where it's domesticated is the area where it's heavily grown now. A problem facing pearl millet is if you look at the places where climate change has affected productivity the most, that part of West Africa is absolutely top of the list. It's bright red in that image. This is a minus 40% impact on agricultural productivity that's been attributed to climate change. So millet, which is already a very hardy cereal because of where it's grown, is facing even more threats. And so the first set of experiments we did with some collaborators from Laurent Laplace's lab in Montpellier was look at seedlings and look at primary root growth. So 16 Passot from Laurent's lab spent a lot of her PhD using rhizotrons and the 2D system I showed you earlier and the first version of RootNav to look at how the primary root develops in pearl millet. And a striking thing of pearl millet is how fast the primary root grows. It grows exponentially speedily till about 6 days after germination. And at that point, the primary root extension rate stabilizes, and lateral roots and crown roots start to appear. But that first burst of growth is, oh, sorry, I haven't got to that, definitely under genetic control. And it begged the question is there an advantage in certain situations to having a primary root that grows so quickly, and why? And if we look at the climate where millet is grown, it's those parts of West Africa, and this is for Senegal, which I'll talk about mainly from now on. It has a defined rainy season where almost all the rain comes. And the way millet is grown, it is traditionally sown just before or just after the first rain event of the rainy season. And that gets germination out of the way. And then we're waiting for the second rain event to really kick off the growth. However, and whether this is linked to climate change or it's just a thing that's always happened, in recent years, there's been a delay between that first rainfall event and the second rainfall event. So in the period 2018 to 2021, 3 of those years, there was a gap of at least 21 days between the first rainfall event and the second. And those farmers that put their millets out after the first rain event or just before it, that's immediately facing an intermittent drought stress for up to 21 days. 2020, which was unfortunately the year we couldn't do a field trial because of COVID, bucked that trend. There was nice consistent rain all the way through. But certainly this is a thing that happens to millet in the field in Africa. You get this intermittent drought early on. And some work by Carla de la Fuente, again from Laurent Laplace's lab, again using the 2D system at Nottingham, looked at millet inbred lines, 122 lines in a panel, and there's a nice diversity in primary root length of seedlings, which they correlated back to primary root growth. They then did a field trial of a subset of those lines that varied for primary root length. And over those 2 years, field performance correlated incredibly well with root length. So they had an advantage in the 2 years of the field trials they did if they had a primary root that grew nice and long. Because this was a sequence line and they delivered a genome by sequencing, they could go gene hunting, and they found a potential candidate. It's a glutaredoxin. They called it GRX-C9, and it appears to work by increasing cell length. And that kind of fits in the story of being a glutaredoxin because they're redox genes. And we know redox is involved in cell expansion in the cell wall. And that was kind of proven by looking at there is no increase in cell production between lines that are slow and fast, but there is an increase in cell length. We can't yet do functional genetics in millet. Groups are working on it, but we haven't got that technologies. But we can, of course, go back to Arabidopsis, and the Arabidopsis homologue of GRX-C9, knock it out, you get a reduced primary root growth. And again, you see exactly the same thing. It's affecting cell length, not cell production rate. So we have good evidence that we have the genetic control in millet for primary root length, and it's this glutaredoxin gene. So on the strength of that, we went back to our funders and we proposed doing a nice long project in Senegal to look at the root system of millet and how it can cover these intermittent drought events. And this was funded by the Royal Society under the International Collaboration Award system. It involves us, CIRAS and ISRA, who are 2 organizations inside Senegal, and again, IRD in Montpellier, who have very strong links to France, to Senegal. And this was how we designed the experiment. We have 2 field plots, one irrigated, one not irrigated. Both irrigated. And we did this in the dry season, so there's no effect of rainfall. And then we imposed a drought by simply withholding irrigation 21 days after sowing in the stress conditions. We imposed that for another 21 days, so at 42 days after sowing, We collected our samples for phenotyping. Then we let the rest of the plots carry on until harvest, where we again phenotyped for yield and yield components. So it's a 2-year study. We lost 2020, unfortunately, again because of COVID. This time we use an expanded panel of 160 fully sequenced lines. We have our 2 treatments, we have 4 reps. So we ended up with 1,280 plots grown in the field for those 2 years. And of those plots, we used a lot of the techniques I talked about earlier in the talk. We did shovelomics, we flew some drones for some temperature and NDVI measurements. We sampled leaf discs for the ionome for ionomics at Nottingham's ionomics facility, and we subsampled some of those shovelomic sections to extract roots to send back to the UK for latting. So this is a summary of everything we measured with drone flights. We have the morphological traits. We did some more genome by sequencing to make sure everything was right with our genetic maps. Biomass and ion contact, root architecture via the shovelomic system, and then anatomical traits via that. And so to give an idea of the logistical problem of doing this at high throughput is, so in 2021, we ended up with 3,000 samples sent to us, which gave us 70,000 images, because we take about 20 images per root, which adds up to about 0.6 of a terabyte. That gets quite difficult to handle, that kind of sheer size. So we end up curating it down to single image sets to a mere 24 gigabytes, and that's what we fed to our deep learning program. In 2022, we ended up with 0.7 terabytes of data. Again, we've processed and sent that all through the system. And here are the results from these experiments. Immediately, the phenotyping of the normal yield traits like biomass, we see a reduction in the imposed stress in both years, quite dramatic. There's also recovery at the end, at maturity, in both years. 2022 seemed to recover a little bit better, but we still have recovery afterwards. But there is a price to pay. So when you've had this intermittent stress for 21 days, you don't recover yield. You might get the biomass back, but you don't get the yield. So again, we have grain yield decreases in both years. So this shows that the importance or the damage that can happen with this intermittent drought event, but also the real impact it has on yield. And the fact that we could actually reproduce it in this experiment, which was done slightly earlier in the dry season, as I said, to avoid rainfall events. So we looked at this data and we calculated stress tolerance indices, which are trying to work out the recovery of lines, how well they were at recovering from this drought event, both at the end of the drought event and at yield. And nicely, and for our experimental design, both in 2021 and 2022, the stress tolerance index at the phenotyping stage, at that 21 days after 21 days of drought, correlates nicely with the yield value for the trait. And we have some other recovery indices we were testing at the same time. More importantly and interestingly for me and us is that of the anatomical traits we measured, metaxylem area jumped out as correlating to a better tolerance index at yield in both years. Nothing else really correlated that well, except for, as you can see in 2022, one of the root angle traits from the shovelomics experiment weakly correlates with yield. So there is some evidence root angle is important, but it seems like in these experiments anyway, the metaxylem is doing something interesting. And luckily, as I was saying, because this is a population, we have the fully sequenced, we fully sequenced it, we can look at the heritability, and it's under genetic control, metaxylem area, and doing a little bit of GWAS, we can start to see some candidate areas where the genes might sit to control that trait. So that's for future work, but we definitely something worth looking at. But the question of what the metaxylem's doing and why it's important couldn't be answered from those trials. So we set up some extra experiments using lysimeters. This is a lysimeter system in-house in Senegal, and Montpellier has instrumented scales in a glass house under a system that works really well for these kind of experiments to look at transpiration rates and that kind of thing and drying down. So we took 12 contrasting genotypes from the study, contrasting for metaxylem size and for the ones that seem to have this recovery. And then we started to look at the link between metaxylem size and transpiration. The idea being that by controlling transpiration rates, you can either take water up longer or not lose it when you have to. And looking at the first set of data that came out from this experiment, transpiration efficiency did nicely correlate with stress tolerance and biomass at maturity. So it's definitely, or potentially, transpiration is what's happening here. And also linking to the anatomical work, transpiration efficiency was correlated with mean metaxylem size and total metaxylem area. So again, we have potentially a link here. It's metaxylem size feeding into transpiration to hydraulics, and that's what's helping them survive this drought period. This is preliminary data from the lysimeter. This needs to be looked at in a few more lines, but the lines with a larger metaxylem area do seem to be able to maintain transpiration in drier soils. So you could imagine this might be a mechanism for conferring the advantage we've seen. If they can stay transpiring longer before you hit a certain fraction of available water, you'll get more water, which will help you survive the period. Or conversely, when rain comes back, you'll be able to move to full transpiration at a lower available water outside. This definitely requires some extra work. We're repeating this experiment and we're looking at the other lines, but this is looking very promising for a potential mechanism linking the metaxylem area that we saw in the field and the ability to survive. So the current state of play of this project is that we're repeating the lysimeter experiment. We have completed the experiment. We've harvested samples for laser ablation from that experiment, and Pablo Aforti, the PhD student who's been doing this work in Montpellier, was at Nottingham up to last week, putting those through the LAT. We're analysing them as we speak, and hopefully that will confirm or deny some of those hypotheses and the experiments we had before me. The story with some of the other traits, like a lot of these when you only do 2 years of field trials instead of the 3 or 4 we'd really like to do, is that the correlations are a bit weak. There are some trends, but nothing that really jumps out. One that we can't quite explain, but is consistent, is that one of the root angle traits, the width of the steepness of the root angle, is correlating to the ionomic profile of potassium, calcium, and phosphorus in the leaves. Not exactly sure how or why that could happen. It might be the area of the soil being exploited by the root system at a particular angle. We're not sure, but that's quite weak, and it only happened in the well-watered plot. But hopefully, when we get the final analysis of this information, the story will be clear, and we'll have a nice story to tell about how these anatomical traits are important in pearl millet in overcoming this intermittent stress. So, coming up to the hour, good timing. The kind of take-homes I'd like to give for this are that roots are important. They're absolutely important. They're a great thing to study. But obviously, they're only part of the story. It's the whole plant. Everything that's happening above, as we saw with the transpiration story, that's got to be affected by anatomy of the stem, anatomy of the leaves, physiological things, all kinds of stuff like that. I haven't talked too much about what's going on in the soil and the soil around the plants and how roots interact with that. That is obviously something that's hugely important, particularly soil structure and how roots interact and change that, and the microbiome, how the microbiome and the plant together alter the soil, how the microbiome might alter the plant, alter the soil. These are also things that should be looked at when we're looking at this kind of phenotyping of roots and soil. On the technology side, there are, as you've seen, a lot of techniques. we still don't have very easily deployed in-field techniques. That's something that I'm not sure we ever will. It's very, very difficult. A few people have tried portable X-ray machines to put in fields, but I don't think that will work. Software's come a long way, and that's helped us turn these large data sets into numbers we can use, so we can actually do decent genetics on it. 3D is still difficult. So a lot of the lat information you saw there was 2D. We can absolutely render that into 3D, but extracting data in 3D is a challenge. And we have some funding to look at that in the future, but that's not there at the moment. And things like risotrons are still difficult to reliably segment root systems out of, because in a risotron, you don't get that clean entire root system against the glass. It will grow in and out of the glass. There'll be soils all around it. And as you saw in the images I've shown, some of the best systems we can use still aren't as good as the others. So there's some work that could be done there. And hopefully some of this machine learning, deep learning programs will help us solve some of those problems. And my big take home of these techniques is they are all complementary. They help each other. You can do a seedling screen with huge numbers, and then you go to a smaller number of risotrons of larger plants, and then you go to a field. I don't think there's one system that you should use all the time. I think you should pick and choose however your experiment requires it. And take homes for the millet story is that there are two stories. One is the seedling screens revealed this story about primary root growth and how the primary root can be important in getting over things like early drought. And it's been a nice test bed for anatomical phenotyping of lat samples acquired in the real world and sent for testing. And it looks like it's pointing us in the right direction to look at some interesting traits for targeted breeding in the future. So with that, I have a huge list of acknowledgements. As you can imagine, this work's been done over many years from very large teams. I have probably forgotten a few people, so I hope if they're not listening in or watching a recording, I apologize in advance. The group in France is led by Laurent and Alex, and a lot of the work you saw today, Pablo Aforti is the PhD student who's leading on the work from Senegal. 16 Passo and Carla de Fuente did the work on pearl millet in the future, and everybody else in that team helped at various times. In Serras and Isra in Senegal, great collaborators. They have an excellent system out there. Gido Kane was in charge of all that systems. At Nottingham, we have two groups of people really involved in a lot of this work. In biosciences, John Atkinson, who designs a lot of the phenotyping systems with me. Malcolm runs some of our root groups, so he knows a lot about what's going on here. Rahul, Dylan and Stuart are PhD students who built various of these systems. Craig and Brian run the Hounsfield, so they're all part of the CT. And over in computer science, Ezzy Benson wrote the root scan software, and Michael Pound, Andrew French and Tony Pridmore are the kind of the leaders of the computer vision lab. We've received funding from a lot of sources. The big funders for us are the BBSRC and NERC, and the ERC funded big programs in the Hounsfield that help some of the systems. And the EPPN transferable access program funded a lot of the visits for the early millet work. And of course, the Royal Society and ANR were funding the big projects in France and Senegal. So with that, I'll end on an advert. This September, we have the second town hall and conference for UK plant phenomics. Rob will be speaking and a guest of honor to tell us all how things have been going up there with you. And we'll be updating on what's going on in the plant phenomics landscape, but we're also going to be obviously canvassing your information and your knowledge about what we're going to do next. Because we're coming to the end of the scoping exercise, and the next thing is to get ready to put in a bid for the UK's national infrastructure. So I believe it's free this year, but you can register at either of those links below. So with that, I'll stop sharing, and I'm happy to take as many questions as you have.
Thank you for that, Darren. Very wide-ranging and interesting presentation. So, are there any questions from the floor? Tim?
Hi, Darren.
Oh yeah.
Yeah, thanks for that. I mean, a really great example of how you can do high throughput work on large populations with roots that has impact. I mean, it's kind of the holy grail, and you've shown that it's possible. One of the questions I had was particularly with the work in Senegal, where you're looking at drought tolerance and so on. I wonder if something like suberin laydown is also an important trait, and whether there's something whether you're able to measure that with your anatomical screening as well.
Yeah, absolutely. It's one of the things that we put on the trait list. As you saw in those millet images, that ring of sclerenchyma is almost certainly suberin lignin. And we honestly thought that would be one of the traits that would jump out amongst these lines, because it is differential amongst that population. We see more suberinization, slack lignification, less. We see them in slightly different places. But it didn't actually pan out as much as the metaxylem. So yes, I think it's important, maybe not so much there, but we can definitely look at it.
Yeah, I guess it's a temporal thing as well, isn't it? Because I mean, suberin might be a longer term fix, whereas if you're looking at very quick changes, then maybe it's not so important.
Exactly, and it might be. So one of the things I liked about this study was it was a real world imposition of the problem they have. Too many times, I think we overdo it. We drought things till they die, and we give them no water. And in the real world, if it was that droughted, you just abandon that harvest and move on to the next one. This kind of 21-day bit of drought is it's quite problematic, because you have traits that don't have time to develop in that 21-day period. So they're not going to be relevant, but they might have other benefits in water use efficiency that makes the line better to withstand it anyway. So I think that's one of the things that's muddied that data a bit. And something like that, the suberinization, might be an overall ability to survive drought rather than the intermittent drought bit. And kind of overlaying those two bits of data gets quite tricky.
Anyway, thanks for flying the flag for roots. That's great. Cheers.
So actually, Darren, that leads into a question that I had. So obviously you're looking at sort of macro architectural traits and some of these micro anatomical traits. Do you see any scope and looking into the future? Is there what are the technologies available that might allow you to start to look into the chemical traits of roots or root exudates or some of these other areas?
Yeah, I think exudates might be tricky, because there aren't great systems to kind of image them. There are better systems to extract them, like microdialysis and capillaries and things like that. So, but that's not quite the same. You're then taking them away and then putting through a mass spec or something. Internally, the imaging is getting a bit better. We've just got another alert bid. Thank you, BBSRC. Times are hard. We need all the funding we can get for a fast lifetime imaging fluorescence system. And so this lets you image with FLIM, which is effectively chemotyping. And what we'd like to do with that is go from LAT images, run them over to that, and then do chemotyping on the image. And then we can start identifying chemical signals inside the root and absolutely saying, yes, this fluorescence signal is suberin, this is lignin. Because at the moment, we're just seeing a strong fluorescence and we're kind of guessing what they are. So I think like that, chemotyping via imaging is definitely doing, and things like nanosims, surface ion mass spec, and imaging mass spec will tell you things. But developing the system to get something kind of realistically and rooty into these machines is the fun bit. But that might be an answer, Sims.
Okay, thanks. Can you have a question?
Yeah, it kind of links back to things that you've already mentioned, actually, and going back to the metaxylem. I was just curious as to whether it was, I'm not sure if you mentioned it or I couldn't pick up what the relationship was between the metaxylem and the response, whether a reduction in metaxylem area or an increase. And I was wondering if it was linked to potential cavitation resistance and the drought resistance.
Yeah, so it kind of, we're not sure. is being completely honest, because it kind of went the opposite way. I was expecting, I think there's a Goldilocks size of metaxylem. Too big and you're going to cavitate. Bigger, you're going to be able to, it looks like you can still maintain a high transpiration rate even when the soil is a little bit dry. So you don't see that fall off in transpiration when you hit a certain soil water level. And that might mean you can suck up a bit more water a bit longer at the first part of the drought period. But the danger is, as you said, you're then going to run into cavitation problems when you get really dry. So I was kind of expecting too big metaxylem to be a problem, too small to be a problem, and in the middle to be okay. But we didn't see that oversized thing as an issue. That might be because 21 days drought isn't enough to reach cavitation size, but that's something it'd be nice to look at in the future, particularly in things like those lysimeters. It's a much more controlled system. If you're using a lysimeter in a glass house, we can probably put cavitation probes on and have a look and see if there are cavitation events happening, because that might be interesting.
Just if I may, with just a little add-on question with that, I wonder,
Yeah, sure.
I'm not a geneticist, but I wonder, but I'm very interested in anatomical stuff. I've already done some stuff previously with Jonathan on anatomical features using that. But I wonder whether you could actually use LAT as a better screening technique and relate anatomical features to function of root rather than potentially looking at genetics.
Yeah,
as it might be cheaper.
Yeah, absolutely. I mean, the beauty of any high throughput system is you produce lots and lots of data, and you can run lots of samples through. And once your sample size gets big enough, the statistics starts to help you a lot more, and you can just run your statistical analysis on. I mean, we're getting to some of these semi-autonomous deep learning-y stuff where you just throw the numbers at it and tell me something interesting about this data, and it might well find exactly that. Yeah, it might not be genetic, it may be functional.
Thank you.
Eric, you had a question.
Yeah, I really enjoyed that. I love the level of detail. It's fantastic. So thinking about phenotyping going upscale rather than down to more detail is trying to phenotype at field scale. So can you see any advances coming in things like ground penetrating radar or electrical conductivity, so you can get a kind of meta phenotyping at a field level?
Yeah, not immediately, but I've been, like you, I've been hoping for it for a long time, because I'm very schizophrenic about this. I love highly detailed, getting all the information out. But pragmatically, I would rather have 10,000 measurements at 80% accuracy than 100 measurements at 100% accuracy, because that's what statistics is for. That then makes it useful. So if there were proxies like ERT that were close enough to what's going on with the roots, the fact that you can survey a whole field in a few hours makes that data much more valuable than 5 cores taken across the same area of field. And you know exactly what's gone on in those 5 cores, because the bane of our lives is heterogeneity in the soil. But in a field, the soil is not the same 1 meter to the next meter. And it's one thing for me to fill 120 pots with topsoil that's all exactly the same because I'm controlling all my conditions, but that's not the real world at all. So yeah, there's absolutely a need. There's a newish technique, a kind of variation on the impedance tomography that's supposed to work in fields, but I've not seen that in action. But ground penetrating radar and ERT, they're not quite there yet, I think. They're good proxies, but okay, so I'm in danger of waffling on far too much here, so you jump in if I do. There's an idea that you can use these different techniques to learn from each other. So you can ERT a cube of soil and get an ERT signal. You then take that cube of soil, put it in a CT scanner and scan it, and then say, here's where the roots really were. Now you give a deep learning program, you say, magic black box, tell me how to get from that to that. And by the way, that is my limit of what I know about machine learning, the magic black box. But we are thinking about this idea of using different techniques to train models on other techniques. And then there might be something that we can't see, but a deep learning program can. And so maybe that's a way of improving things like ERT and EMI, of actually throwing all this power we have now in deep machine learning at them and trying to get them more accurate that way.
I'm sure there's already an AI spin-out company being formed as we talk, Darren.
Almost certainly. And
Thanks very
I
much.
won't be able to afford their services, so it won't be a problem.
Okay, do we have any more questions for Darren? Can't see any hands coming up. So I think then we'd just finally like to thank you again, Darren, for a really interesting and entertaining talk.
Oh, thanks.
And to remind people that I believe we have another seminar in 2 weeks' time. So please feel free to join us then. Thank you again, Darren.
Oh, great. And I look forward to coming up and seeing everything in action.
Absolutely. Thanks.
Thank you.